All right, welcome everybody. This is really wonderful to see so many of you here at our next installment of the SLAC public lecture series, which we hold every couple of months or so. I have to tell you a little secret that to me, the most fun part of being on a committee is the fact I'm learning science that I know nothing about. And this is another example I'm actually learning from Macon about very interesting aspects of biology, chemistry, and so on and so forth, which is not necessarily my own area. I'm an astrophysicist by training. So without much more ado, Macon is here working in the group of Dr. Beatty, who is sitting right in front of here, which is a group of structural and molecular biology group. His career started from undergraduate work at Western Washington University, I believe, near Seattle, north of Seattle, probably closer to Vancouver. And that was the beginning of your work on sort of, if I understand correctly, on the effects of silver nanoparticles on algal growth, right? And then you moved on to University of California, Riverside, where you started to work connection between metals and organic molecules. That brought you exciting and growing group. So thank you very much for being here. Thank you.
Yeah, thank you. Thanks, Greg. All right. Well,
we will take questions in the audience by raising your hand. You will hear more about that. And people on Zoom actually will not be able to do this. I know that there's actually quite a connection here, quite a number of people who are dialing in, but this lecture will be recorded in the case if you want to actually watch some subtle aspects that you maybe feel like. All right, sorry,
perfect.
Go ahead.
Okay, good. And you can all hear me all right? Good, good. Okay, well, thank you so much for being here. It's really my pleasure to have been asked to give this lecture. And I'm really excited to share 3 stories with you all tonight. So thank you for the introduction, Greg. So like I said, I'm going to share 3 stories with you all tonight. Each story is fairly disparate from the other. So they might not seem quite connected at first, but you'll quickly find, I hope, that they are in fact connected by something that takes a little bit of motivation to drive interest in, and that is a metabolic pathway. And we'll get into what exactly that pathway is. I can almost promise you've never heard of it, and what exactly its significance is over the course of the next 50 minutes or so. So just to follow up on what Greg said, I was asked to put in a little bit of personal background. I am indeed from Washington State. I highly recommend visiting there, especially this time of year. The leaves are changing. It's absolutely gorgeous. And I did my PhD down in Southern California in the Inland Empire, so maybe not the beach coastal part of Southern California that you typically think of. And you know, coming from Washington State, we don't have any palm trees there. And so moving to somewhere that had palm trees was quite shocking. They are very alien looking. If you're not used to, if you're not from somewhere that has these kind of trees, they're very strange. And in other ways, Riverside couldn't be more different from Seattle. But I had a great time down there. And perhaps most importantly for this talk, my time at UC Riverside introduced me to doing science here at SLAC, specifically at the Stanford Synchrotron radiation light source, which is one of 2 accelerators, 2 large accelerators that we operate here on site. You see, as I was, I should say, I did my background is in actually soil science of all things and kind of biogeochemistry. I was looking at how toxic metals in ground and in soil and sediments kind of transform as they react with different mineral phases with an eye on groundwater quality, human health, and these kinds of things. And we were able to use the X-rays at the Stanford Synchrotron Radiation Light Source, or SSRL, to study that. And doing experiments here at SSRL is kind of an ordeal. You know, you're only given a set number of days any given year that you can come and do your science here, and you can take your measurements. So it's very precious, which means you're doing it 24/7, you know, for however many days that you have. So you're going to go a little loopy, and, you know, you got to kind of keep yourself awake. And so, you know, on more than one occasion, I'd be pacing around this building. It's a circular building. You can kind of see a general layout here, you know, 2 or 3 a.m. trying to keep the juices flowing. And I'd come across these different posters, which are all up on the inside of the building. And in fact, you know, there are posters kind of everywhere on this campus. And this poster in particular caught my eye. It's still hanging up, which is kind of cool. And it was some work done, I think, on hemoglobin by a scientist here at SLAC named Riti Sarangi. And she, I thought she did some really cool work. I didn't know her at this time, but I was thrilled because around the end of my PhD, an email came across on one of the listservs that I was involved with, and she was looking for a postdoc. And I applied and had the great fortune to be accepted into the group, and I'm excited now to share with you some of the science that we do here in the structural and molecular biology group at SSRL and at SLAC. So I'm going to take you back to the beginning. We're all familiar with this. This is, of course, the planet we live on, our pale blue dot, mostly blue, some green, some brown, and depending on the time of the year, perhaps a lot of smoke. But the young Earth was very different from this planet that we live on today. It was different in so many ways. It was primarily an ocean world. At this time, the oceans were roughly twice as deep as they are today, which is quite surprising. You might ask where that extra ocean's worth of water went. It actually was taken up into the rocks of our planet, and a very important process to the history of life that I'll get into. But before there were oceans, Earth was kind of quite an unpleasant place to be. And I've got this artwork here. You might recognize this. I actually saw this artwork in one of my middle school science books about 20 years ago. And at the time, we thought the young Earth during the Hadean, which you can see right here, the Earth is about 4.5 billion years old or so. The Hadean was the first, let's say, 500 million years. For the longest time, we thought that during this first chapter of Earth's history, this is what the Earth looked like, an absolute hellscape, noxious fumes, a terrible atmosphere, pelting down asteroids. It was a really unpleasant place to be. But I'm thankful to say that in the 20 years it's been since I've been in middle school, scientists have been hard at work chronicling and reestablishing and refining our understanding of what Earth looked like. We now know that these oceans that were twice as deep as our oceans are today, they actually formed much earlier than we thought. They formed about 4.3 to 4.2 billion years ago, which is astounding. That means the Earth cooled off and things stabilized enough that liquid water could form. Of course, the atmosphere had no oxygen in it at the time. And depending on which geologist you are going to engage with, there's some thoughts that plate tectonics had actually started by this time, or at least a nascent form of what would come to be plate tectonics, which is going to play a really important role in the story of the origin of life on Earth. And then I actually had to amend this slide just a few hours ago because I came across a new study that was only published a month ago that kind of pushes this back. But previously, the earliest known signs of life that we have, like concrete evidence of life on planet Earth, date back about 4 billion years ago. And then I crossed this study this afternoon that pushes that back a further 200 million years. So now it looks like life may have popped up very shortly after liquid oceans formed, which is kind of astounding. So we have a very small window then, where this planet, which is an ocean world, went from being sterile to being covered in life to such an extent that we can detect traces of it today. So if we want to understand what this life was like, it really would be beneficial to us to understand a little bit more about the geology of the planet as it was at the time. So I've already talked about the oceans. That's all fine and good. But there's another thing. You see the atmosphere of this young planet had no oxygen, had little to no nitrogen, probably, at least as far as what I was able to tell from reading the literature. But what it did have in its atmosphere was CO2, and it had a lot of it. Upward estimates that I was able to find suggested upwards of 10 bar, which is 10 times our current atmospheric pressure, but essentially all CO2, quite a lot. And the oceans of this young Earth were doing the same thing that our oceans are doing today. We're all familiar with climate change, more CO2 going into the atmosphere, and luckily our oceans buffer us against some of that, the effects of climate change, by absorbing CO2 into the water, right? Perhaps you've heard of that. This same phenomenon was, of course, happening on the young Earth, and with so much more CO2 in the atmosphere, there was much more CO2 in the ocean. So to tell you this next part, there were two different ways in which I could go about telling you what I'm going to tell you. The first is using subduction as an illustration. And again, subduction perhaps was taking place on the Earth at this time, perhaps not. Another way that I could phrase it is with a spreading ridge. This is a region on the planet where new crust is forming. We have one of these straight down the middle of the Atlantic, and I'll show you that in a little bit. But for the sake of simplicity, I think a lot of us are more familiar with subduction than we are these spreading zones. So we're going to go with this. So on this planet, when you have these tremendous volumes of rock, that make up our crust, and at this time, most of the crust would have been this basalt, this ocean basalt. There likely would have been little to no continental crust, which is higher in silica, it's less buoyant. But on today's world, of course, we know that ocean crust subducts under continental crust, and this has to do with the differences in their densities. And perhaps something similar was happening on the early Earth. Perhaps it was a related phenomena. But in either case, you have these tremendous volumes of rock exerting tremendous force on each other, inducing cracking, faulting, fissures, and the like. And this happens today, of course. And on an ocean world, if we induce these cracks into this ocean basalt, water will, of course, infiltrate. And now there's something really special about basalt. So basalt is a rock, and it's composed of many different minerals, but most of the minerals in basalt are found in the olivine family. These are silica-poor, iron-rich rocks. And that's what makes them so dense. They're chock full of iron. And what does iron do when it comes in contact with water? Well, on today, of course, it rusts. Back then, there was no oxygen to cause the rusting, but it turns out that water will react with olivine and essentially rust the olivine. Through a process called serpentinization, water reacts with the olivine to form secondary minerals like lizardite and other serpentinite minerals. And these kind of take olivine, which is this lovely green color down here, to a slightly less lovely green and yellow, brown, I suppose. And this process of water incorporation into the rock is critical. It releases tremendous amounts of gas in the form of hydrogen, I should say hydrogen gas. It also releases a lot of sulfur and other transition metal species, ions, and the like, as olivine is essentially transforming itself into this new structure. Here I'm showing lizardite. There's one other product of this reaction, though, that's crucial, and that is the release of heat. If you take a cubic meter of olivine, so a cubic meter of basalt, and react it with water, let it have as much water as it wants, it will end up absorbing about 300 liters or 600 pounds or so with water. And in the process, it'll release about 600 million joules of heat, which is enough heat to power the average American home for about a week. And that's only from 35 cubic feet of rock. So you can imagine if you're talking about a young planet with nothing but basalt and nothing but water, this is quite a powerful reaction. And even today, this reaction is ongoing. And we're all familiar with, I don't know, a mug of tea or coffee or boiling water. And when you apply heat to a fluid, like water or coffee or whatever, you'll induce convection, right? And convection is this turning of a fluid to induce temperature, normalization across the body of the fluid. And the same thing would happen at depth with this water, now chock full of hydrogen and sulfur and whatever. And this convection would carry this new hot water back up to the surface of the ocean. Now, there's something I failed to mention at this point about what this ocean water was like. This ocean water was slightly acidic. I mean, we're kind of familiar with that today, right? We've heard that there's like coral bleaching and stuff with, as climate change kind of goes on, the oceans seem to be growing acidic. Well, the same was true of the young ocean on the young planet Earth. It was mildly acidic, roughly pH 5 to 6. This isn't so acidic that it would affect you. You could drink it and be fine. But the hydrothermal vent fluid, in the process of reacting, I'm sorry, I'll just call it vent fluid for now. In the process of reacting with the olivine, it's essentially had its protons stripped out. And in fact, those protons are what is forming the hydrogen gas. And so it's mildly basic. It's about the same pH as would be water that you would go, specialty water, specialty alkaline water that you buy at the store. And something fun happens when you mix this vent fluid with the ocean water. You start precipitating copious quantities of rock. And this rock is, of course, calcium carbonate. It's the same rock that forms hydrothermal vents today. It's the same rock that, of course, forms the shells of coral and mollusks and other things in our oceans. And you get hydrothermal vents. And so these vents that I'm talking about are quite different from the vents that you may be familiar with. This is, of course, a black smoker, which were discovered in the 1970s. And I know you all know this. They belch out these billowing clouds of black smoke. They're kind of scary looking, and they exist in the depths of the ocean. Well, these vents don't exist quite as deep in the ocean. They were only just discovered about 25 years ago by Dr. Deborah Kelly. She's a professor at the University of Washington, go Huskies. And she actually took the Alvin down, if you remember the name of that sub, and she found the first installment of what would turn out to be an incredible complex of these vents down at the bottom of the ocean. And since then, we found many more. And we now know that based on the conditions of the early Earth, the geologic conditions, these kinds of vents would have been incredibly, would have been incredibly widespread across the seafloor. And we also know, there's some precedence for the ancient age of these vents. This is actually a fossilized alkaline hydrothermal vent. This was found in Ireland, of all places. It dates back to only 360 million years old, which of course is much younger than the 4 billion year old vents that we would need to push our story forward. But it carries the idea that this phenomenon has been happening on Earth for a very long time, right? Okay, so let's zoom in on one of these vents. This is actually the vent that Dr. Kelly found in 2000. It's in the middle of the Atlantic, and it has a nice cross section to it. And if we kind of zoom in on this vent and we take a closer look, we'll see some startling things, some very unique attributes. So this is a whole vent down here in the center. You can see it almost looks organic, kind of like a coral. It's got all these little tendrils kind of spindling off, which is very nice. But if you were to break off one of those little tendrils, maybe about ½ an inch big, and you were to cut it cross-section wise, and you were to look at it under a microscope, you would see this incredibly intricate geometry on the inside. Now, this is key for the origin of life. At least I like the idea for this hypothesis of how life may have progressed. So if you zoom in further on this intricate geometry down to the micron scale with your microscope, you'll see that not only is it incredibly channelized with these small channels and pores, but it's got these micron-sized compartments throughout the structure. And this is unique. The black smoker vents, they don't have this. So what might this have to do with the origin of life? Well, a couple things. And I'll get to these aren't my ideas, but there's some key folks that I want to credit with this once I talk to you about what one of these compartments are capable of. So if we consider one compartment in the structure of this vent, and we consider that there's this hydrothermal fluid kind of circulating throughout, it's chock full of sulfur and hydrogen gas and dissolved, dissolved metals and ions. And we consider that just microns away through the wall, the wall of the vent, there is dissolved carbon dioxide. There's a lot of dissolved iron and nickel and cobalt and other metals in the early ocean. I don't know when these were actually discovered, but sometime in the recent, not too far past, these great banded iron formations were found. And what we understand now from these great banded iron formations is that the ancient oceans were full of dissolved iron, which I'm showing up there, and that they remained full of dissolved iron until cyanobacteria would come along a few 100 million years after the period of time that I'm talking about, and they would start cranking out oxygen as a waste product. And that oxygen would go on to react with the iron in the oceans, crashing it out to form these red banded iron formations, which are chock full of hematite and other iron minerals. And in fact, they're a primary source for mining iron in a lot of parts of the world. But at this time, cyanobacteria won't be around for a very, very long time. And so there's a lot of iron and nickel in water. And so iron and nickel, they don't only like to react with oxygen, they're actually perfectly happy to react with sulfur as well. And it just so happens that there's plenty of sulfur in this vent fluid. And so, and this is actually what we find in these vents today. It turns out that these vents are coated in kind of a dusting of these nickel iron sulfides like greigite. And these nickel iron sulfides have this very particular shape in their internal mineralogy, a basic unit. And it's these nice cubes of iron and sulfur. You could imagine one of these sulfurs being substituted out for another metal like nickel or cobalt. And these cubes in the form of greigite or these other hydrothermal vent minerals have this uncanny ability to catalyze the formation of organic molecules from CO2 and hydrogen. Now, this is exciting, right? These molecules that I'm showing here are molecules that biochemists would recognize as being key metabolites, key metabolic intermediates for cells, for us, for for bacteria. We all kind of share these small molecules as the key intermediates in our metabolism. Now, this idea of, you can imagine then I should say this, that these compartments, would then keep this organic matter in contact with the greigite, with the sulfide minerals, and they can continue to undergo these reactions, growing in complexity over time. Now, this idea is not mine. It was first proposed by Michael Russell, I believe in 1994, could be 1993. And it's being championed by a few people today, including Nick Lane, who's a professor at University College London. He's got a lab there. And he's done a lot of really good work showing that indeed these vent minerals actually do catalyze these reactions. And he continues to show that these vent minerals under these conditions in the absence of oxygen can catalyze the formation of increasingly complex biologically relevant molecules. And so this is kind of a far cry from an actual living cell, but it might suggest that these kind of vents, this kind of a geochemical system may have been the cradle for life on Earth. And unfortunately, this is where I'm going to end this portion of my talk. And if you're interested in learning more, I would highly recommend either watching some of Nick's lectures online. He is a really good speaker, or finding his books. He's got quite a few books out there which are written for an audience who may or may not have a background in science. I've certainly read a few of them, and they're very good. So I've proposed this vent system that can very importantly take carbon dioxide and hydrogen and turn it into these metabolically relevant molecules. Great, now what? Well, it turns out that there's one metabolic pathway, and only one that we are aware of, that can take hydrogen and carbon dioxide and fix it into these small molecules, specifically acetate. And you all know what acetate is. Probably put it on your salads. It's vinegar, right? And acetate is a very small molecule. It's 2 carbons. One of those carbons has 2 oxygens on it, and one of those carbons has 3 hydrogens. I just drew it as a stick. And this is remarkable. We only know of 6 carbon fixation pathways. No other pathway accepts hydrogen and carbon dioxide as inputs, let alone under anaerobic conditions. And I should say it's called the Wood-Ljungdahl pathway, if I haven't said that already. And if you take a look at the proteins, which are key to the Wood-Ljungdahl pathway, you'll see that they are replete with iron sulfur clusters. Iron sulfur clusters turn out to be really important in biology, in a lot of other proteins. And I can go into that maybe in the Q&A section if you're interested, but for now, it's sufficient to know that these proteins just have these iron sulfur clusters everywhere within them. But can we do better, right? All I've done so far is shown you that, okay, so this chemistry can happen in vents, and this guy in London can like replicate this chemistry. And hey, look, it kind of looks like this pathway that we know of. But can we do better? And we can. And something that's cool about working in this field is I could not have given this talk like 7 years ago, because what I'm about to show you has only just come out. This came out in 2018, and then a more recent paper earlier this year. So if you're a biochemist and you're interested in knowing how old a protein is, there are a few things that you can do, but typically what's done is you find the gene that codes for that protein, and then you go to the oldest domains of life. You go to the oldest families of organisms that you can find, and typically this is the bacteria and the archaea. Bacteria, of course, single-celled organisms. We all are familiar with them and make us sick sometimes. They help us out at other times. And archaea are like bacteria. They're single-celled organisms, but the way they do their chemistry, the way they replicate their DNA, the lipids they have in their membranes are very different from bacteria. So we know that they're divergent from each other, and we know that they came from a universal common ancestor, or the last universal common ancestor. And so if you go out there as a very determined biochemist or geneticist, and you survey thousands of bacteria and thousands of archaea for the genes that you're interested in this case, the genes that code for proteins in the Wood-Ljungdahl pathway, what we find is that these genes are everywhere in both bacteria and archaea, which is fascinating because they branched off from each other, and they branched off from the last universal common ancestor billions of years ago, upwards of this is actually an old number now. This is from 2018. This has been updated this year to 4.2 billion years ago when the archaea branched off. And anyway, so you find them everywhere, which, via inference can tell you that these genes must have also been present in the last universal common ancestor to all life on Earth that we are currently aware of. Right now, we're only aware of one universal common ancestor because we can essentially find the commonality among all life that we know of, right? There aren't these 2 separate branches, although it's possible that LUCA, our universal common ancestor, had years of its own, whose descendants didn't survive until the present day. Okay, that's enough about the past. We're still going to be in the past, but I'm going to take you forward now 4 billion years, 4.5 billion years, I suppose, to the 20th century. I'm going to introduce you to a young man named Ernest Lawrence. This is the mid-1930s. Ernest was a professor at Yale University. He'd only just started there when the University of California came knocking and was like, hey, you should come work for us instead. And he said, okay, I suppose the West Coast sounded all right to him. You might recognize the name because he has a national lab now that bears his name, LBNL. Well, shortly after starting his tenure at Berkeley, he would go on to patent and create the first particle accelerator. This was a cyclotron. It was a device that could fit in the palm of your hand. It was 5 inches in diameter. Or I'm sorry, yeah, diameter. So, you know, very human-sized. And what this thing would do is it would accept ionized hydrogen gas as an input, and it would basically spiral that hydrogen gas using magnetic and electric fields on the inside up to tremendous speeds, and then shoot it out in a straight line. You know, that could have a lot of applications, and certainly they thought so over at Berkeley, and they would go on to build larger and larger implementations of a cyclotron, and they found out that if you shot these particles at a target, and this could be a target, you know, they tried a lot of material. It could be a target of graphite. It could be a target of ammonium nitrate, which is explosive. I actually did this. And you would bombard it with these particles, you could create a new form of matter. And this form of matter, of course, is radioactive carbon, among other things. You can make other forms of matter this way too, but they made these radioactive isotopes of carbon, carbon-11 and carbon-14. Carbon-11 has a half-life that's really short. It's like 20 minutes, so it'll be gone super fast. Carbon-14, half-life of 5,700 years. It's the form of carbon that, you know, folks talk about when they talk about carbon dating. And these forms of carbon would be really important to the discovery, the human discovery, I should say, of the Wood-Ljungdahl pathway. So around the same time, around the exact same time that Ernest is first drawing up his patent for the 5-inch cyclotron, a microbiologist over in Europe by the name of Wieringa, and I probably said his last name correctly, discovered the first organism that could live on just hydrogen and CO2. And this was remarkable. It could produce acetate as a byproduct. And this was very exciting. You know, nothing had ever been found that could live on such meager rations before. And typically, yeah, and then it was lost. And this was a bummer. This was a real bummer because this, again, the only known one, it was published 1936 and then lost from what I can tell about 4 years later. Luckily, it would go on to be found in the 1980s and recultured. It was literally in this like dusty old test tube in the back of this guy's lab who had died, you know, a long time ago. It was a whole thing. But that's okay. You know, science doesn't wait for people to find things in test tubes, and which was a real bummer, right? And the reason it's a bummer is biochemists typically, when they find something new, like an organism that can grow off of hydrogen and CO2, they really like to just stick with that one organism until they understand everything about that pathway, right? I mean, you don't want to be confusing yourself by going and looking at all these other things. And so it's really unfortunate that that organism wouldn't become the model organism for the Wood-Ljungdahl pathway. Instead, this one would. This is an organism that we now call Morella thermoaceticum. I don't have the name up there because you don't need to remember that. It was discovered shortly after, in fact, right around the time that first organism was lost. It was found. And it was found not to live on hydrogen and CO2, but to ferment glucose into acetate. Later, it would be found that it could live off hydrogen and CO2, but that would come much later. It's at this point that I want to talk to you about acetate again. So, you know, I showed you acetate before. It's like a small molecule, right? So then what is this guy? Well, this is acetyl-CoA. See, acetate is a really small molecule, and it's typically chaperoned around the cell by this much larger molecule called coenzyme A, which is you know, quite a bit more complex, but it acts to kind of ferry the acetate to exactly where it needs to go within the cell. It's actually quite a remarkable design. Anyway, when I talk about acetate from now on, this is the form of acetate I'm talking about, but you can still think of it as just 2 little carbons with some oxygen attached to it. So it's at this point that you might wonder, yeah, okay, so what? Like, you know, these acetogens, these acetate producing bacteria seem all right, but like, what's the big deal? Well, aside from their implications with respect to the origin of life, which we now know LUCA was almost certainly an acetogen. And that came out this year. That's brand new information. These guys are everywhere. And because they're so ancient, they run away from oxygen. They will die in the presence of oxygen. And so they live everywhere on Earth that oxygen isn't. This includes our own digestive tracts, cows, the digestive tracts of insects. I read that they're so abundant in the digestive tracts of termites that people think termites might live purely on the acetate that these bacteria produce as their main source of carbon. So they really are everywhere. They're incredibly widespread. There are many different types, and they've evolved over the years to have many different metabolic pathways now. So some of them don't even solely rely on the Wood-Ljungdahl pathway to produce acetate. They've interacted with other bacteria, and they acquired other ways of sustaining themselves. But they also do some services for us. With climate change kind of raging, these bugs managed to remove about 100 million US tons of CO2 from the lower atmosphere every year, which is great. It's really nice. That helps us out a little bit. They also produce about 100 billion tons. Yeah, that number's right, per year of acetate. And if you do the math between, these two numbers, you might notice that CO2 number is a lot less, about 1,000th of this acetate number. And so you might ask, where's the rest of the CO2 coming from? I'm pretty sure it's coming from the ground. CO2 is produced at depth in soils and sediments and whatnot. And these bugs do us the favor of consuming that CO2 before it can reach the atmosphere and contribute to climate change, which is great. So at this point, now that we kind of are familiar with what acetogens are, let's go back to Berkeley and let's meet this guy, Martin Kamen and Horace Barker. Martin was actually the guy who discovered carbon-11 and carbon-14. Horace was a microbiologist. They would go on to study this now model acetogen, Morella, and they would be the first to use these new radioactive isotopes of carbon that Martin had just discovered, carbon-14 in particular. And they would find that if they incubated this microbe with glucose and carbon-14, you would get radioactive acetate, which was the first time this had ever been seen, at least in this organism. Quite remarkable. Around the same time in the US, just as it is today, the US was prolific in research at the time. Out in the Midwest, out in Iowa, there was this young lad named Harlan Wood. He began his PhD in the midst of the Depression. In his autobiography, he talks about how grateful he was because he had a wife at the time, and they were really strapped for cash to get this position as a graduate student because it paid at least something while so many struggling. Not to say he didn't struggle, because grad students don't make a lot now, and I can't imagine they made much more then. Anyway, his PhD advisor gave him this task of investigating these bacteria called propionic acid bacteria. That's fine. I mean, I don't know about you. I've never heard of these. It turns out I actually care about these bacteria a lot. They're the bacteria that are responsible for cheese production and the fermentation of other things that we eat, like sauerkraut, right? So, highly important. He would also go on to discover a few other things that challenged the dogma of biochemistry at the time. His research advisor wouldn't let him put those findings into his PhD thesis, which is a bummer. And then someone else discovered the same thing a few years later. And this thing in particular that he claimed to have found was the first known instance of heterotrophic carbon fixation, which is to say an organism that, like us, has to consume carbon. It's not a plant. It can't manufacture its own carbon from CO2. And yet these bacteria, which need to eat carbon to survive, were found to incorporate CO2 into their cell mass. And that was the first time this was ever found. Unfortunately, another group would make the same finding a few years later and publish it. That's how science goes sometimes. Now, Harlan would go on to have a great career. He would continue to use these radioactive isotopes to study metabolism, particularly here. He would be in some sort of race with the researchers over at Berkeley who were also investigating metabolism using these methods. He would go on to have encounters with the US government and the Manhattan Project, who at the time had commandeered the lab of one of his friends to produce uranium. And this really upset Harlan's research because he relied on that friend to produce for him a certain type of carbon for his own research. And that was just a whole mess. Now, a few years later, there we go. Oh, there we go. Good. So a few years later, about a dozen years after that first paper came out where Martin and Horace fed these microbes radioactive carbon dioxide and observed radioactive acetate formed, Harland, he's kind of a professor at this time at Case Western University, and he would take on this Swedish grad student by the name of Lars Ljungdahl. Now, Lars was a little bit older when he started his PhD. I think he was close to 40. And he basically had a whole career before coming to work with Harland. And specifically, he had a career working as a microbiologist in a brewery in Sweden, which is pretty cool. We don't typically think of beer plants as places of science, but this one that he worked at certainly was. And because of his experience working in anaerobic systems and looking at metabolites at this brewery, Harland was like, hey, you should tackle this other really challenging system where we're trying, we want to understand why this acetate is formed from carbon dioxide. And, Lars, being a grad student, agreed, and this would begin a long collaboration that would last until Harland's death, and then Lars would continue working on this system until his death, where over decades, they would refine their understanding of what would come to be known as the Wood-Ljungdahl pathway, that is the fixation of carbon dioxide and hydrogen into acetate. And now I'm going to show you how their understanding of this pathway grew in complexity over decades. You don't really need to pay attention too much to the details here, but you can notice that over time, their understanding of how this happened grew in complexity, and it continued to grow. And much effort and many grad students and postdocs went into developing this until we arrive at the Wood-Ljungdahl pathway as we understand it today. Sorry. Now, the Wood-Ljungdahl pathway, as we understand it, is composed of two halves. There's one half which accepts a carbon dioxide, and this carbon dioxide kind of spins through the cycle and eventually is turned into a methyl group, which is simply one carbon with a few hydrogens attached to it. It's a methyl group in the form of methyl tetrahydrofolate, which you might know better as vitamin B9. This is a vitamin that we require in order to sustain ourselves, and our bodies can make some of this stuff. We still have to eat like folic acid. Eat your greens and get your folic acid, and it'll turn into methyl tetrahydrofolate in your body. That's great. And during this process, they made some notable discoveries. I just threw this one in here because I thought it was so cool. They found the first protein in nature that actually used tungsten in its active site. I mean, I don't know about you. I don't encounter tungsten every day. I thought this was pretty cool that there's this enzyme out there that's crucial to the production of vitamin B9 that uses tungsten. And then there's the carbonyl branch. The carbonyl branch is the branch that I personally work in. It's the area that we're involved in. It's composed of 3 proteins in the branch. There's a carbon monoxide dehydrogenase, or CODH, which takes carbon dioxide, again, as an input, and basically rips it apart into carbon monoxide and water. Then there's, oh, I'm so sorry. Let it all play. All right. Oh my God. Oh no, that's fine. All right. Try not to waste too much of your time. Anyway, then there's the carbonyl branch. And while I let this play, I'll talk you through these other proteins. So the carbonyl branch, aside from carbon monoxide dehydrogenase, also has a CFESP, which is a little ferry protein. And what it does is it takes the methyl group from that vitamin B9 that I showed you, and it ferries it over from the methyl branch to the carbonyl branch. So it's just a shuttle. And it delivers that methyl group to this other protein, acetyl-CoA synthase. And acetyl-CoA synthase takes the methyl group from CFESP, and it takes the carbon monoxide from CODH, and it combines them together to form acetate. It really does this remarkable synthesis. And this really is unlike any other carbon fixation pathway that we're aware of. At this point, that I'll introduce our final character of the night. This is Steve Ragsdale. He's our collaborator. He was a grad student of Lars, and he did his postdoc with Harland. He knew the two men very well, and he speaks highly of them. So they were both very friendly, good people. Unfortunately, of course, they're no longer with us. And he's been working in the system for a long time. He's made many contributions, some of which I'll get into here. But first, let's take a look at these proteins. So I've mentioned ACS and CODH, which are involved in the acetate synthesis. And it turns out that these are actually typically fused into one larger protein. And if we take a look again at what this protein has in it, along with CFESP, although I won't be talking very much about it, you'll see that they're chock full of these iron sulfur clusters, these iron sulfur cubes. And if you hone in on the active site where the chemistry actually happens, you'll see that not only do they have these iron sulfur clusters, but there's nickel there. And the nickel turns out to be very important. And I'll get into that. I'm not going to talk about CFSP anymore. So I've kind of shown you the Wood-Ljungdahl pathway, how CO2 is turned into this acetate. That's great. What possibly more, what more is there to know? And where can synchrotron light come into the picture? Well, it turns out that, if you really want to know something, you've got to ask very nitty-gritty detailed questions. So for CODH, how exactly does it take carbon dioxide and turn it into carbon monoxide? What are the painful steps that it goes through to make that happen? Or for acetate, how exactly are the carbon monoxide and methyl group mushed together to form acetate? What are the agonizing steps that the protein goes through to make that happen? And that is where synchrotron light comes into play. So the process of measuring samples at SSRL, especially delicate biochemical samples, is somewhat of an ordeal. First of all, we have to partner with a very talented biochemist. This is someone who has the expertise in expressing these proteins, which often are very oxygen sensitive. And we have to go through all this step of, these steps of shipping and freezing, and we load them into our cryostat where we measure them at just above the temperature of outer space at one of our many beamlines. Each of them has the capability to, hit our sample with X-rays, and we have 3 here that I'm showing you. Each beamline allows us to measure a slightly different thing or a slightly different aspect of our sample. All very useful. But all of the beamlines are roughly the same. Again, pathway for the X-rays to come through. We've got our cryostat where our sample is being kept cold, and then we've got our detectors where we can measure the X-rays that come off our samples. And during the process of doing this, we can have happy times. If our samples are really good, if our collaborators did a really good job prepping our samples, we'll be happy. We can have questionable times where we see kind of a confusing signal, and occasionally we can have pretty rough times if our samples turn out to be not prepared correctly, which can happen. But in the end, we're always able to collect some really good data to tell our stories with, to do our research with. Before I get into the final little bit of this talk, I did want to mention just what exactly X-ray absorption spectroscopy is, because it's what we do after all, and it's how we're able to tell all the things that I will show you soon about these systems. With X-ray absorption spectroscopy, we use the synchrotron, we use the different beamlines to tune X-rays to a very specific energy. And this is the energy that binds the most tightly held electrons of our element of interest. So say nickel is our element of interest. The most tightly held electrons with nickel. And when they are hit by these X-ray photons that have been finely tuned, those electrons get ejected and promoted into a much higher energy state. And if we keep doing that incrementally with higher and higher energy X-rays, we can collect a spectrum that looks like this. And this spectra can tell us a few different things depending on which part of it you're looking at. If you look at this first part, you can get detailed information about the electrons in your system. And this matters a lot because how these electrons are kind of structured around your metal will dictate the reactivity of the metal towards a carbonyl, towards a carbon monoxide or a methyl group. If you look at this section and you do some math to kind of magnify the wiggles that you can barely see in that flat line, you get a nice sinusoidal wave, which will tell you geometric information, what atoms and how far they are from your central absorber, which is quite nice. Kind of like throwing a rock into a pond. Bounce off the metaphor that's usually used for this. And you get something that looks like this that'll give you some nice detailed molecular structure. And that's great. So how can we use these techniques to study ACS? Well, we can ask very specific questions like, where does the carbonyl bind? Does it engage with those really important iron sulfur clusters? Or does it bind to the nickel? What's going on? And these are the kind of questions that we can ask. And indeed, Reedy and Steve did ask these. Before I came on, she partnered with Steve to ask some questions about acetyl-CoA synthase. And this is the active side of acetyl-CoA synthase. You'll see there's an iron sulfur cluster up there, and it's attached to 2 nickels. So we had to ask the question, I'm giving it away a little bit here, but which of the nickels does the carbon monoxide bind to? It turns out that when you expose acetyl-CoA synthase to just carbon monoxide, you get a very different signal from the protein that hasn't been exposed to carbon monoxide, which is great. We love big signals. They make the data processing really easy. And when this data is analyzed, Reedy was able to tell that the carbon monoxide binds to the top of the middle nickel, kind of forming a little pyramid or tetrahedron. Whereas in the absence of carbon monoxide, a little water comes in and binds to that nickel, but not to the top, rather to the side. So you form kind of this little square, right, which is kind of cool. I came on later and we wanted to ask the same question, but about the methyl and acetylated forms of this protein using XAFs and these other X-ray absorption techniques to do so. And we were able to tell that unlike the carbon monoxide, the methyl group actually displaces the water and binds where the water would bind. And then fascinatingly, the acetate seems to be at the same position in the corner of this square plane, which, you know, via inference means that that carbon monoxide is kind of swinging down and inserting itself into the bond between the methyl group and the nickel. So there's some interesting chemistry that's happening here that we can only tell via XAS. And I should say at this point that this validation of carbon monoxide binding to the nickel or the methyl or the acetyl groups binding to the nickel, these are the first times we've ever seen directly these intermediates of the Wood-Ljungdahl pathway. Before this, it was, you know, suspected, highly suspected, that the Wood-Ljungdahl pathway progressed through these direct metal-carbon bonds, which is very unlike any other carbon fixation pathway. For example, the Calvin cycle, which is what plants use to fix carbon, doesn't do this. It doesn't progress through organometallic intermediates. So this was a big deal. We can also ask questions about the role of the nickel. So just to backtrack a little bit, Nick Lane and his lab has done some work that showed that in the absence of nickel, if you only have iron sulfur minerals, hydrothermal vent minerals, they are incapable of catalyzing CO2 and hydrogen into small molecules. But as soon as you add nickel, the reaction proceeds. Again, I'd refer you to his work if you want to see it. It's really approachable to read. But we can ask some detailed questions about, okay, what exactly is the importance of the nickel then? Why is the iron and sulfur so important for this? Well, it turns out the story is electronic. So what I'm showing you here is a kind of an MO diagram. The red clouds are one electron and the blue clouds are a second electron. And we understand electrons as quantum objects, which means they can kind of experience a probabilistic delocalization over a set of atoms, right? And it turns out that these iron sulfur clusters are really good at wicking away electron density or giving electron density to the nickel. And the nickel is incredibly important because it is able to adopt a variety of charge states and conformations, geometries and whatnot, that are required to react with the different intermediates. So it needs a different charge state and geometry to react with the carbonyl, with the carbon monoxide, than it does to react with the methyl group, for example. And it's able to access all of these geometries only because of the iron sulfur cluster. Next, we wanted to ask some similar questions about carbon monoxide dehydrogenase. And you'll notice the active site of this protein is very different from what I just showed you with the ACS. It kind of looks like a cube, but it kind of looks like a cube where one of those irons has been plucked out and swapped with a nickel, but the iron wasn't completely plucked out. It's kind of just dangling here, right? And that's really interesting. That's unique. And quite frankly, we struggle to recreate this ourselves in labs. So if we want to understand the mechanism about how this weird looking pseudo cube affects carbon fixation, we can basically ask a few questions. For one, we have to hypothesize what such an intermediate might look like. And for that, we've got this kind of little circular diagram up here. You don't really need to look at it too much. I just want you to know that this circular catalytic cycle is a hypothesis. And each of the states in this catalytic cycle is a state that we can test using X-ray absorption spectroscopy. So this protein is really unique. It kind of can exist in this kind of resting, sleeping state. And in order to become active to a state where it can actually interact with carbon monoxide or carbon dioxide, it needs an electron given to it. And as soon as it accepts an electron, a big conformational change happens that we can see with XAS. One way to think of this conformation is that that nickel and that dangling iron come closer together. And this turns out to be really important. In order to engage with CO2, this cluster will bind CO2 in two ways. The nickel will bind to the carbon, and the iron, that dangling iron, will bind to one of the oxygens. And then with the help of some of the surrounding amino acids, they will effectively rip that carbon dioxide in two. And one of the oxygens, the one bound to the dangling iron, will go off and become water and kind of drift away into solution. And then the carbon with the other oxygen attached to it will stay bound to the nickel as carbon monoxide and then eventually diffuse and go over to acetyl-CoA synthase to be turned into acetate. This is quite interesting. So from here, there are a few places that we'd like to go. The first is we want to ask the question, these proteins and these organisms are very oxygen sensitive. Is there anything that can be done to make them less oxygen sensitive? And I apologize for that. I got one of my. This is a relatively new question. It was recently discovered in the last maybe 7 years or so, 8 years, the late 2010s, that there's a form of this protein that can somewhat resist oxygen damage. It can restore itself after being affected by oxygen. And there's a company out there, and actually there's a lot of interest in using these organisms to effectively consume the CO2 from industrial processes like steel production or what have you. Have that CO2, instead of going to the atmosphere, piped into these big vats of acetogens where they can be captured by the Wood-Ljungdahl pathway and turned into acetate or other valuable chemical feedstocks. And then from there, I don't know, those can be sold, and then we've prevented all the CO2 from going into the air. And there's this company, Lancetech, who's actually doing exactly that. And we work with one of our collaborators does work with them, and I think they're a pretty impressive company what they're doing. But a key, it would be really nice if they didn't have to scrub the gases that come out of these industrial factories so much to make sure there's no oxygen in them, right? It would make it cheaper and easier to do. So we'd like to investigate that. Also, we are really curious about how this big chaperone fits into the active site to steal the acetate group away from this center nickel. I mean, that's such a large molecule, and the active site around this protein is, I mean, it's kind of big, but it's not that big. So how does that work? These are questions that remain that we really we want to look into using the synchrotron techniques that we have here at SSRL. With that, I want to thank my collaborators. One person I didn't name is Kareem Abulson. He's a friend of mine. He is a grad student of Steve's, and he's done tremendous work in the carbon monoxide dehydrogenase sphere. And with that, I am happy to take any questions.
Right, wonderful. Thank you very much for the great lecture. And I think we have time for questions. So please raise your hand, and I think one of us will deliver my.
Yeah. Oh, here, do you want to take the microphone?
How did you get from acetate to animals like cows, dogs, mammals, humans, fish, etc.? How, in summarization, how did animals come from acid?
How did animals come from acid? Acetate. Oh, good. So acid, I don't know about dogs coming out of pits of acid, but acetate, that's a great question. So once you've taken carbon dioxide, right, which is a gas, it doesn't do us any good. And you take it and you put it into a structure, even as small as acetate. There are other pathways within the cell that can take this acetate and they can complexify it. They can combine it with nitrogen and phosphorus, turn it basically into the proteins and fats and everything else that we need. So it's a pretty complex process. And we can't, I mean, we can do something kind of similar, but we mostly need to eat all of that stuff. Yeah, that's a really good way to think about it. Yep. Yeah.
Can you go back?
Yeah.
One, I believe.
This one? Here?
No, one more. Yes, this one. Have you considered the conformation changes
In the
and
protein?
it becomes more like a spiral, condensed spiral?
Yeah. Yeah, it's really interesting. I mean, the ACS-CODH complex undergoes some really substantial conformational changes because it actually has to directly interface with CFESP to accept the methyl group. And I didn't mention this, but CFESP, which is that methyl ferry protein that brings the methyl group from one branch to the other, actually has vitamin B12 at its active site. That's what ferries the methyl group over directly. I mean, it sounds like you know something about ACS-CODH. Okay. Well, ACS-CODH basically has to undergo this tremendous conformational change at the active site to accept this other huge globular protein in, bringing that methyl group into very close contact with that center nickel, the proximal nickel. As far as those other specific changes that you were talking about with respect to acetyl-CoA, which is not shown, no, I have not thought about that, but I will now. Thank you. Yeah. Oh, yes, very good. Yeah. All right.
Yes, I'm just curious, from all the processes that you've described,
Mhm.
I'm just thinking that here we are, this third rock from a sun that was once a glowing mass of what, rock and
Yeah.
molten rock. Isn't it logical to think that this similar processes could happen on other planets and other solar systems and other moons?
Yeah, it's a really good question. So astrobiology is a huge deal. I think NASA's Jet Propulsion Lab actually has a whole group dedicated to studying not only the astrobiology part, of course they have the astrobiology part, but they actually have a group that actually studies this, investigates this event question. And Nick Lane, who I'm just going to keep referring to because I really love his work, has referenced their work. And I actually was just reading a paper published by the NASA folks before this, and that is an active area of investigation, specifically with respect to Enceladus, right, which is this ice, a moon of Jupiter, right, that has these geysers that are coming out, and those geysers happen to be rich not only in carbon dioxide and hydrogen, but also in some other very suspicious-looking organic molecules. So that's something that, you know, they're evolutionary biochemists and astrobiologists, and I am not, unfortunately, but I'm aware of their work, and that is, yeah, exactly what they're. It's very possible. Yeah. Yeah.
Iron sulfate helps the nickel bond to the carbon. But are there other transition metals that can perform the role of nickel, or is nickel the only interesting one of the hydrothermal?
That's a really good question. And I feel like you're teeing me up here because this is something I didn't have time to include in my talk. Understanding ACS and CODH has been an incredibly painful process. I mean, I just showed you how it essentially took 100 years to get to where we are now. But actually in the early 2000s, I want to say 2003, 2000 and 2004, the first crystal structures of ACS and CODH together came out. And in those crystal structures, they actually had copper or zinc in that middle position. And so for a while, we were thinking that it was copper or zinc that was the catalytic species. And it turns out not to be the case. Those are actually contaminants, which will stall the protein and make it inactive. You'll get a nice structure, but it's not quite catalytically competent as the dinickel species. And as far as CODH goes, I'm not actually sure about substitutions into CODH. And then as far as vent minerals go, I do not know the answer to that either. So I'm sorry about that. Thank you. Okay, time to go. Yeah.
Very nice talk.
Oh, thank you.
So I don't know too much about chemistry, but I do beamline experiments. And I was wondering how much this process that you're studying depends on the ambient environment. So you're doing experiments at 10 Kelvin in a vacuum chamber. This is very different than a hydrothermal vent. So does it matter, is my question.
Oh, that's a really fair question. So a couple things. I mean, sounds like you work on beamlines. So we keep our samples at 10 Kelvin because otherwise the X-rays will just scorch them. Yeah, absolutely, they'll destroy them. The actual kinetically competent, you know, measurements where you're looking to see acetate production in cells, this is all done in labs, you know, under, you know, sometimes basically under the conditions under which these acetogens live. Not all of them live under vents, you know, some of them live at temperatures and pressures that are more amenable to humans. And so with that respect, the organism that we work with a few, but Morella that I introduced is one, Clostridium hydrogeniformans is another. These are organisms that are really well characterized. We understand the environments that they live in, and we understand what their ideal culturing, like habitat, I guess, would be, to which they can, you know, produce acetate at a good rate. So with that being said, we have a great deal of confidence that these measurements are actually indicative of what are going on in the cell. Yeah. Oh. Yeah, with this, are you talking about like AlphaFold or something like that? Well, actually, the man there holding your mic is our resident AI expert in our group. This is Wei Ying He, and he's an AlphaFold expert helping us out right now with another system. And so, yeah, I expect AlphaFold to be very useful, especially for proteins that are perhaps not this one. It turns out that ACS and CODH actually give pretty good crystal structures, so we can understand them structurally in that way. You know, I mentioned this paper that just came out this year where these folks were showing that, oh, actually the last universal common ancestor was likely an acetogen from like that kind of genetic phylogeny ancestry experiment. They actually had some AI, or I should just say machine learning techniques in their methods, right? And so it already is being very useful. But as far as the structural determination goes, I do think so. And Wayne is helping us with that. You. Yeah. Oh, yeah. Oh, yeah. Oh. No, that would be really great. There should be. I just looked over towards my boss and she looked away. There should be. You know, we mostly work with Steve, and we have another collaborator, Kathy Drennan at MIT, who I think was the first one to get the crystal structure of ACS. She's not an exophile person, but these are kind of, you know, we have a small group of people that we're working with right now. You know, it's one of those things where there are so many great systems that we could work on, and quite frankly, we're all very passionate and we'd love to do it. We just need to maybe get some more people, more postdocs and RAs to help us do that. But I'm interested, yeah, especially if it'll help us shed light on these processes that are ongoing. Yeah, oh, I agree, 100%, yeah, for sure. Thank you. Night is young. We can keep going if you guys want to. Yeah. The what one? The planet? Oh, with the hydrothermal vents? Oh, the Earth. The first Earth was a recent geospatial satellite image taken a few weeks ago that I found online. And the other one was, don't hate me, but it was an AI image of trying to, you know, craft an early Earth, because I don't have that much money and I can't pay an artist to make one, especially on these short deadlines. Yeah. Yeah.
I'm an absolute child in these matters, but I do believe I remember from quite some time ago that Darwin, so going back a long time, had the belief that the gentle forcing between day and night and temperature cycling that comes with the day and night somehow or other contributed to the formation of early life. Is that in any way consistent with what's going on here?
I think it is. So there's another group. So I only mentioned Nick Lane because he's kind of prolific and does great writing. There are, of course, many other people working in the origin of life space, and they have a lot of really good theories that they can back up with data about how life would have formed. One of those groups, last name is Page, and I'm really sorry, I can't remember the first name. They also really like this vent idea, but they have taken it in kind of a different direction. It turns out that there are also these shallow alkaline hydrothermal vents, which kind of form in mud flats. There are some of these vents in Australia, and what was the other one? I've already forgotten. But at least in Australia. And these vents, you know, they are shallow. And so they literally form in these tidal flats that not only receive daylight cycles, a diurnal cycle, but they experience the temperature changes and the tidal changes of the surface as well, including becoming completely drained at low tide. So they're exposed to oxygen. And so there's a great deal of interest in the geochemistry and potential biogeochemistry in the sense of like the early life that could have happened on those vents. And I just want to add to that, it's very unlikely that we would see any new forms of life happening in those vents today because we have so much oxygen around us now. It's really inhibitory to this kind of process.
Maybe at this point we exhausted all the questions. So
Okay, great.
let's thank Macon again.
Well, thank you for coming. Yeah, that's true. We have most of our little SMB group here. So if you have questions, there's a lot of us who can chat with you. So thank you for coming. Have a good night.